So it looks like the sample's about 50 grams. It's a little bit more. I took some out to do the soil microbe testing. I'm going to do a aggregate size sediment test. I'm not sure if I'm really going to get any gravel because this is mostly like pine needles and duff. I might not even get any rocks at all. So then I'll get an idea that even the organic matter should sift out, but we'll see how this works out. It looks like it's almost completely organic material. So I'm going to take this 50 grams and mix it up. I'll probably need 2 cups of water in there, and I'll let you know what the measurements are and show you the rest. So just to give us a rough idea of the density, I put this in my jar, and it's about the volume of about 6 fluid ounces. So even though it has water and all these things, we can still get an idea somewhat of the compaction of the soil. And if we were to dry it, we'd know how much water was in there. Here I have some filtered water from a brand new Brita. The one on the right's actually pre-filter and the one on the left is post-filter. I'd probably rank both of them around a 6.5, but if I look closely to my eye, actually, and I did it a couple times, the pre-filter, just the regular tap water, is a little bit more acidic than after it's filtered. Might be contrary to what you might think, because we usually have hard water sometimes here, but they do use aluminum in the system, so it could be because of that. I don't know, but it looks, good enough to go on. It's slightly acidic, and it gives me a baseline when I do the pH after I get some soil extract. So I used 12 ounces of filtered water and put 50 grams of the soil material in and shook it. So what should happen is if there are little rocks in there, they'll sink. This is sort of a different sample than what I was thinking when I realized I could do this test, because it's all floating and it's all organic. So we'll get what information we can from this, and I'm also going to get, I'll filter off a little bit of this water, and I'll do a pH test on this in about 20 minutes too. So as I moved it over here to hopefully be undisturbed, I was able to see like some rocks in the bottom and a little bit of sand. So I'll be able to see a little bit of that, and we'll see how everything else settles out. Now I'm setting up the nematode test. I have about 1 gram of soil. I'm going to wrap it up with the paper towel, fold it, put it in the plastic bag, and... Okay, so here we have my nematode trap. The soil is wrapped up in the paper, and there's some of the filtered water in there. And as you can see, it goes down to this little corner. Got a little piece of soil in there, but anyways, in the corner, what's going to happen is the nematodes will be able to wiggle out of the mesh of the paper towel, and will want to because it's all wet and they're drowning, and then they'll actually free fall through the water because they can't really swim, and they'll all collect excuse me, down at the bottom of this little corner here, where I can pipette them out and put them under the microscope and maybe tell what kinds they are. So it takes a couple hours or even overnight for them to come out, and I will get other microbes in the fluid. Some will be able to swim, and some will collect down there with the nematodes. So I wasn't able to connect my camera to my microscope like I had hoped, but a lot of these pieces are really stuck together, and it makes me think that they might be full of fungal hyphae. So I was going to take a look under the microscope and just see if I can just visualize anything by taking a look. super hard to get this on camera. It's like being unreasonable. But there are some tiny filamentous little, you can see, I can right there in the middle, see that little white crooked line, the small one, that's mycelium holding it all together, breaking down all this humusy wood duff. That's what it looks like to me. Sorry for making you dizzy. I have to hold the light too. I don't have a dissecting microscope. I have to put the light from above. So here's just a little liquid. I use this straw to pipette out of my solution that I'm getting to precipitate. And so it's been settling for about 15 minutes. And I just pipetted it out with the straw. I'm going to do a couple things. I'm going to do just a pH test just with the strips just right now. I'll probably do that again like tomorrow when I actually look at the sediment. And then I'm going to put a drop of this on a slide and see what's in there wiggling around. So of course a lot is left to the eye with the litmus strips. Actually this one, although it's really not long enough, we'll leave it for a lot longer. And the lighting is not really showing it, but it actually is looking closer to a 7. Much close. It's not quite a 7, but it's significantly a little bit more basic than the original distilled water was. We could do a comparison, just test the distilled water again, so I could hold it up right next to it. But my sense is it's actually making a little more alkaline. It's still super close, basically hasn't done anything. So just a couple tiny little drops. Probably a whole universe is in there. Let's find out. There won't be any nematodes because they tend to hang out in the soil. That's why we have to do the special nematode trap. But we'll get other microbes. So this is what I'm looking at. I think I'm at 140 mag. I have to check that. $160. Okay, so not a whole lot of guys moving around. I did see 1 or 2. I think they're probably there. You see a few, something Euglena, maybe. Have to look a little closer. Single-celled organisms, and then we have lots of little flecks of like humus-like particles and some fibers of cellulose. I would think seems. I don't know if you can see in that light, but quite a few little green things. Almost looks like algae, could just be little bits of moss, like that still kind of have some chlorophyll in there. Not teeming with life, actually a little bit still, a lot more still than you would see like in garden soil, for instance. Found some places with a little more moving around. I still want to call a lot of them Euglena. You can see some of those like perfectly round little, probably algae guys, I would think. What is interesting, you can see these like some of the fibers are more clear. You can see it moving. This is not, I didn't put a cover slip on because I wanted to watch them move around and interact. Those little fibers like around 230 up in the right hand side that are like clear, I would say that those are mycelium, like broken off fragments of mushroom vegetative tissue. The other fibers are probably more like wood pulp fibers, I would think. And some of them are clearly moving of their own volition. Here we have a lot more of the, you see all those like specifically circular round little dots. I would think that those are algae. Oh, there's a wiggler. I have to get in a little closer to really identify these guys. But those little circular dots could also be like an ascomycete, like like a unicellular fungi. This could be the very helper guys that the orchids need to live. There's some more hyphae over there, the clear. I think, if I really knew my stuff, I could look at like the clamp connections and tell what the hyphae was, but that's what genetics are for. But this is an interesting shot, I think, because you can, the more I look at them, I don't see any red pigment or chlorophyll in them, the more I think that those maybe are little just unicellular fungi or spores they could be. Look at those guys, must have little cilia propelling them around. A whole universe is in there. It's so fun. Oh, I gotta get this thing connected to the computer so that you don't have to watch all the wiggling. Maybe I'll let the slide hang out. So now I'll let it sit so that it's been like about an hour from when I first mixed the soil and water, and then I'll let that wait until tomorrow to see the sediment that's come out and see if I can draw on my geology knowledge from 20 years ago and see if I can identify any of those rocks. So 50 grams for 6 ounces gives us a bunch of calculations later. 4.8 grams per cubic inch. A rough calculation, because I just kind of tapped it in the jar, but it's still useful. So I did another pH test, and I'm getting the water from, you can see maybe how a lot of the like organic debris is suspended, and there's a little bit of sand at the bottom and gravel maybe, and there's like fluid. I know it's hard to see here, but and there's like that water layer in there. So that's where I got the water, and I looked at the microbes and also where I got the water to check the pH, and the pH really showed just no change. I think that this soil ends up having a fairly neutral pH, even though the high amount of like forest stuff made me initially think it might be acidic. But I think what ends up being is that it's sort of inert. It's not super actively decomposing. It's like higher carbon and less nitrogen. So if I were to take a stab and a guess at the cation exchange capacity, it would be fairly low. So the soil is kind of makes more like a media, which would, I mean, to turn it into cultural terms for orchids, then you're making it's like a substrate that provides structure for the roots and a place for nutrients to pass through, but the soil itself, the substrate itself, the media doesn't actually have a whole lot of nutrition. So if you think of using like coconut coir and vermiculite or perlite to grow orchids in instead of like a soil-based media, that's kind of what I'm seeing here is like a lot of this forest stuff that's really not very decomposed at all. A lot of that brown stuff was like lignins, not instead of cellulose, and then some sand, a little bit of sand down there in the bottom. And tomorrow I'll take a look at that sand. And also the nematodes, I might take a look at the nematodes later tonight, but I'll probably give them until tomorrow. They might not be as active if I don't get to it after work, but I did take a look at the corner of the bag, the nematode bag, and I don't see any in there yet. Sometimes when there's a whole lot of them, you can actually like see them with your naked eye. So I'll show you that later. Here's the bag, just so you can take a look, and you can see there's like a little bit of kind of tea colored forming from there's some humic acid or tannins dissolving, or possibly from some microbes coming through. But so right down in that corner is where they're gonna gather. Oh, there we go. If they are there. And I'll be able to tell if they're like plant parasitic nematodes or saprophytic nematodes or like carnivorous nematodes. Potentially be able to tell by looking at their mouth parts. If I find any, I'll have to look very closely.